let's go. Graduated, elevated, now it's my time to shine, yeah, let's go. Graduated, elevated, now it's my time to shine, yeah, let's go. Major.
Hello and welcome to PhD Hard Talk. I am your host, Noma Mguni. Welcome, Jordan. Could you please introduce your research? No, you're fine. Your research and also who you are and if you're in first year, second year, and then we'll crack on from there.
Excellent. Hi, my name is Jordan Barrows. I'm a 6th year now. PhD student at Johns Hopkins University. I work in Erin Goley's lab studying bacterial cell division. So we work with a couple of different bacteria. The one that I'm work most with is one you've probably never heard of. It's a cute little bug named Caulobacter crescentus that lives in the wild in ponds and in soil. And it's a very useful model organism for studying how bacteria go throughout their lives and how they progress. So we are using this to understand how one bacteria becomes 2 bacteria, which is an essential part of any organism's life. In order to live and survive, you have to be able to divide and propagate. And we've known quite a bit about this process for a very long time. And there are several different players that are involved, but a lot of the actual roles of each of those components is still kind of a mystery. And so we are leveraging genetics, microscopy, and biochemistry to try to answer questions about how bacterial cell division is taking place, how it's getting initiated and regulated, and then what perturbations we can make to it that will affect it, and so on and so forth, and how it responds to different cues in the environment.
Amazing. So I know that there's bacteria, and I've just come to learn that we've got little things living on our bodies as well.
It's a harrowing lesson, isn't it? Yeah.
Yeah. So I'm quite interested in your research, because obviously it's something new, and I think to anybody that's sat at home, you wouldn't really know about bacteria like that. So I'm looking forward to our discussion today. So I guess first things first, what motivated you to want to look at, bacteria for 6 years?
I mean, bacteria are awesome. That's why. I've always kind of had a fascination for bacteria. In high school, I was able to work with viruses that actually attack and kill bacteria specifically. They're called bacteriophage, and they look like little alien spacecraft. If you haven't seen them, you should Google bacteriophages, and it'll blow your mind. So they infect bacteria. And they're very specific. They don't harm any other organism. And then throughout the years, I've just always thought bacteria were the coolest thing since sliced bread. And I really like that they are very adaptable to their environments. And so, humans and other larger organisms, we have thousands of different types of cells that can all perform different duties, and we act as one organism. But a bacteria is just a single cell on its own out in the wild, and it has to react to whatever life throws at it. And bacteria are not unique in this respect, but they come in all different types of shapes and sizes and different responses to the world, and that just completely fascinates me. I also just really like looking at tiny things, and so bacteria are a perfect model for doing something like that. And when I got here to Johns Hopkins, our PhD programs include 3 rotations in different labs at the beginning, and I knew I wanted to look at bacteria, but what I really fell in love with was the lab that I joined, or ended up joining. And actually, coincidentally, I'm wearing our lab shirt today for a totally other reason, but hey, that works. And the first day in the lab, I knew that was where I wanted to spend the next 6 years. The people were amazing. My boss was super enthusiastic and brilliant and just brought life to science. And so the bacteria were one part of it, but also the personnel and the environment that I was working in is what really drew me in.
Okay, so for anybody that's out there, you know, we hear a lot of things when it comes to labs. So you've just heard something positive today, so we have to take it and we have to embrace it. You know, there is community in labs. It's not that solo. And if you've got somebody, you know, leading that's very personable, it can make you stay for 6 years.
Absolutely. Yeah. I mean, I would stay for another 6 if my boss would hire me for 6 years.
Well, we're throwing that to the universe, boss. So in terms of your research then, I guess there's a word, and I can't pronounce it. I tried to learn how to pronounce it, which is the cytoskeleton proteins.
Uh-huh. Yeah.
What are those? Did I try?
Yeah, you nailed it. So this is the cytoskeleton. So cyto is the Greek word for cell, I think, and then skeleton speaks for itself. And so in the cells like you and I have, this is literally the skeleton of the cell. So there are different types of these components, but they give the cell structure, because the cells in our body are very malleable and flexible. And the protective layer has the consistency of about mineral oil. And so you can imagine that those would not be very structured, or they would not be able to stand up against pressure or things like that. And so the cytoskeleton acts as kind of this internal structural system that helps the cell, helps support the cell, as well as give it shape and things. Bacteria are a little bit unique in that they have a cell wall, in addition to the outer layer that our cells have. And that actually provides a lot of rigidity. But then inside the cells, they also have this cytoskeleton. And rather than provide support for the cells, because that role is performed by this hard, rigid cell wall, these actually will serve as scaffolds for processes within the cell. And so, for example, in order to divide, bacteria, a lot of bacteria typically divide near the center of their cell somewhere. Okay. And so they need to bring all of the machinery that will actually divide the cell to that place. And you can think of it kind of like a construction environment or a construction site. And so, you know, in order to actually build something, you need to bring all of the people there and all of the machines and the cranes in order to get something done. And so the role of the cytoskeleton in bacteria is actually to form a scaffold that signals to the rest of the cell, hey, here is a construction site, and we need to bring all of the other components that are going to be actually carrying out the construction. And so the cytoskeletal element that I study will find the spot where division will occur, and then it will recruit all of the rest of those components, and then actually direct their function. So you can think of it kind of like a construction foreman. And so it's bringing all of the pieces together and then telling them what to do so that the job gets done right and the bacteria doesn't explode.
Oh, wow. That is quite fascinating. I guess I wouldn't necessarily think of bacteria in that way. And also in terms of, you know, the awareness, really, you know, in terms of us being normal people in this instance, we wouldn't really think of bacteria being this flexible and also this, I think I can use the word intelligent.
Yeah. Maybe not in the classical sense, but they're very responsive to their environment, and everything is controlled. It's not just a random series of events, and it's really a beautiful process.
No, it sounds amazing. And I guess, you know, in terms of your investigation, going to methodology, then what is your chosen strategy for your methodology?
Yeah, so that's a great question. So I like to think of the analogy, you know, the best way to understand how something works is to break it.
Okay.
And so if you want to understand, you know, how a computer works, you want to take it apart, you want to turn things off, you want to break something and then figure out what happens. And the approach is similar in a lot of biology, and bacterial biology is no exception. And so one of the things that we can do, assuming that we can perform some genetics, is to actually remove some of these components that I've described. And so this cytoskeletal element, if we remove it, bacteria have two kind of growth modes at the very simplest, or at least rod-shaped bacteria do. So one is that they elongate and they get longer, and then the other is that they divide periodically. And they are more or less independent of each other in the type of bacteria that we study. And so you can actually remove this element that I'm talking about that's controlling the whole process of cell division. And the cells will continue to elongate, and they'll just get longer and longer and longer, but they will never divide. And so you've now, by removing that, we've established that component is essential for division. And so that's where we can begin. And it turns out that a lot of these players that are involved in this process, not just the master regulator, but also some of the components, the things that it controls, are also essential for this process. And so if you remove them, again, the cell division is not able to occur and the cells elongate. And so one of the first things that people did to start studying cell division was they randomly mutagenize bacteria. And you might have, you hear about mutation being associated with disease and with cancer and with other types of problems in the cell. But mutation can also lead to adaptation. So mutation is the basis of evolution, and we don't have to go into that. But basically, if you randomly create mutations in a cell, a lot of things can happen. The most common thing is that you're going to break something because you're going to mess something up and impede its function. And so the very first studies were randomly introducing random mutations and seeing that the cells were no longer able to divide. And then they can see where that mutation occurred, and they found that that was controlling these cytoskeletal elements. So from there, then we can, once we've identified all of the pieces that are playing a role, so, at our construction site, we've identified all of the individuals, we've identified all the equipment. Now we can start to understand how they work. And so we can do some finer genetic experiments and break it in a more fine-tuned way, and then we can start to understand how that works. The second thing that I actually love doing is microscopy, and so a really great way to study something is to be able to see it. And as you can imagine, we need some very nice equipment to be able to see things that are too small to see with the naked eye. In addition to that, we can also make these factors that I've been mentioning, we can attach a little, essentially a little trailer to them or a little tag to them that will make them glow. And so not only can we study the effect of breaking these components, but we can also look at where they are in the cell in real time. And so I can use my microscope to see not only the cells, but also where the internal components of the cells are, and then watch what happens. And then I can change things either about their environment or remove some other components and see how that affects where they are present in the cell, and then how that affects the downstream events like the division eventually. And then finally, as if that wasn't enough, we can actually remove these components from the cell and study them in the test tube. And so the cell is actually a very complicated environment. And so sometimes when you want to understand something, you need to remove all kinds of variables and look at it in isolation. So we can actually take this cytoskeletal element out of the cell and put it in a test tube, and then do a bunch of different tests on it, and to look at both its function and its structure. And then we can even look at that under the microscope as well and see how it looks just on its own. And so a combination of these three methods can give us some insight into how these components are working, and then let us form some hypotheses about how the overall process is working. And then it's kind of, you just repeat that process.
Over and over again.
Yep. Until you've figured it out completely, which is basically never. But we're always getting closer. There's always more to
each year. That's quite fascinating, and I'm thinking, and I'm trying to understand, your work and how it benefits the wider community. So if we understand bacteria a bit better, how does it benefit you and I?
That's a really good question. And so I think the primary motivation in a lot of people's minds is to be able to fight against diseases. So I've mentioned that Caulobacter lives in the environment. I forgot to mention that it is not a pathogen. And so I don't have to wear, big hazmat suits or anything to work with it. So it doesn't infect humans. But the advantage is that we can use this and use what we've learned in organisms like Caulobacter and apply that to other organisms that are pathogenic and that do cause disease. Because there are a lot of similarities between these different organisms. There are also a bunch of differences as well. And so we are not alone in studying bacteria. People are studying all kinds of bacteria, from Neisseria to E. coli to Salmonella to Burkholderia to all kinds of other ones, and tuberculosis, etc. And so we've actually been able to identify things that are common among the bacteria as well as things that are different. And all of that helps us learn more about all of the bacteria that we're studying. And you might be asking, well, how does this help? And so a couple different things. One is that, our primary mode of defense against bacteria is antibiotics. And these are special drugs that specifically target some kind of process in bacteria, be it the protective cell wall or other parts of the life cycle that it needs to live. And these days, antibiotic resistance is becoming very common. Again, this is the problem with things that like to evolve. Bacteria are very, very good at evolving and getting around things like that. And so we really need to find some new strategies for doing this. And so understanding how bacteria work at a very intimate and detailed level is very useful for understanding not only why our drugs are working, but also why they aren't. And hopefully we'll be able to inform us on how to develop better drugs. Beyond that, though, and I actually am really glad that you mentioned the bacteria that you learned are all over you. Not only do we have bacteria all over the surface of our bodies, but we also have them inside our bodies. And so this is in our mouth, in our throat, in our stomach, sometimes, and in our intestines, are what is called the microbiome. And this is actually essential for our life. If you lose your microbiome, it's very difficult for you to survive. They provide several different mechanisms of support for you. So the very first thing is they actually protect you from harmful bacteria. So you have a bunch of bacteria in and around you that are defending you from these pathogenic bacteria. They also help you digest food. So our body is actually not amazing at digesting food. It does okay. But the bacteria in our gut actually do a much better job. I shouldn't say it better. They are able to digest things that we can't.
Yes.
And then they release nutrients and we can use those as well. And so there's actually this intimate connection between us and bacteria that a lot of people are totally unaware of. And we are only just beginning to understand the very complex interactions between us and the bacteria in our bodies, as well as between the bacteria and the other bacteria and how they interact. And again, Caulobacter is an environmental microbe, and so it's not directly related to that, but all of the things that we understand about Caulobacter can be applied to other things. And then finally, in the wider environment, things like Caulobacter live in the wild and they affect things that go on around them. So there are a lot of bacteria that are essential for plants to be able to grow. They form this symbiosis, and much like us, they provide plants with nutrients, and then the plants return the favor. And so things like Caulobacter actually have been found near in rivers and things, and there's some hypotheses that they might have some interaction. And then there's lots of other bacteria that are being heavily studied in those environments as well. And so the more that we can learn about things like Caulobacter and other bacteria, the more that we can start to understand how all of these processes work. That will hopefully help us to get a better understanding of and an appreciation for the role that bacteria are playing in our lives and then be able to leverage that and help keep us safer and more healthy. And not only us, but also the world.
Yeah, I mean, they do say knowledge is power, and I guess from what you shared there, I wouldn't know necessarily on a normal day. So thank you so much. And I'm guessing, I've done some research as well on the internet, and I did find that some bacteria is used to preserve food. And I thought that was fascinating. So could you enlighten us? Because I'm thinking there's someone at home going, oh my gosh, you put bacteria in my milk.
I mean, so I'll be honest, I don't know much about it.
That's all right.
What I do know is that, things like cheese are a combination, and yogurt are a combination of bacteria and fungus cultures that come together to make cheese and yogurt and those types of things. And I'm sure that they play some kind of role. I don't want to say anything more than that because it's not my field of expertise.
It's not your field, yeah.
But I mean, yes, that's definitely a thing that's there. Bacteria can also be used to produce drugs and medicines as well. And so beyond just the food aspect, and also into the health and the medical industry. Not only can they help protect us from pathogens, but they can produce compounds that help us in all walks of life as well.
That is quite interesting. And I guess, you touched on the antibiotics. So is in terms of future then, are you leaning more towards, medicine, or are you, well, food is not your thing. So what does the future look like for you?
The future is never set in stone. I really like studying, we call it basic biology, which is the asking of fundamental questions and then trying to understand something about biology that doesn't necessarily have a direct clinically relevant route. And so something like Caulobacter, where it's not a pathogen, it's not in the hospitals or anything, but we can use it and take advantage of its unique features to study a basic question. And that's what I'm very passionate about, is understanding biology and understanding how these processes work. And so that's more or less the direction that I'm going in. I also, I'm a little squeamish, so so medicine isn't really for me, and I've known that for a long time.
Yeah.
But everything that we learn has some kind of direct connection or indirect connection to that. So likely not on the clinical route for me, but there's always some interplay between those things. And honestly, 10 years ago, I wanted to be an astrophysicist. So in 10 more years, things might be completely different. Who knows?
Right, John Hopkins. So anything is possible, right?
I'm humble enough to know that I am not certain of anything in my life. So.
Thank you very much for your response. So the next session literally is just a quick discussion based on what's been coming in and what's been asked.
Okay.
And in terms of advice, I've received an email which has asked basically in regards to grades. With is it SATs? What do you call them there?
For college or graduate school?
For college.
Okay, yeah. So for getting entrance into college, grades in high school and the SAT and ACT, the standardized testing, those are big factors. Sorry, I'll let you finish your question.
Yeah, no, I was learning, so I'll remember that for next time. So someone wants to go into Yale, basically. Is it Yale University?
Sure.
I should know this. No idea, forgive me. So they've got a standard in terms of what's required. What's your advice to them? Because they've looked at their grades currently and they're not sure. And they're from the global south, and they want to migrate across to the US. And you're currently in research, you're in university, you understand what's going on in terms of inflation or recession, whichever word they're using at the moment, I don't know. And living crisis. What's your advice to them to say, are they okay to stay where they are, or should they come across the pond? What's the advice there?
Boy, that's a very good and difficult question. I will say that I don't have a great perspective on the university, the undergraduate university system as it is right now. The last time I applied to college was a decade ago. And so just bearing in mind that things have changed. I do know for myself that back when I applied, grades and the standardized tests were very much a part of the college application process. I will say that for graduate schools, we've actually dropped the standardized test as a piece of admission because it's been shown to not be a good indicator of success, at least at the graduate level for our types of programs. And I've heard rumors that colleges are also starting to abandon that as well, but do not take my word for that because I'm not sure and I haven't followed up on that. I will say this. So there's this concept of, that you need to get into the most prestigious university possible and that will launch your career. And all of that is certainly true. And having the name recognition behind a university and having all of that will definitely help. That being said, the quality of education at other universities is not lacking. So I didn't go to an Ivy League university. I went to a state university, the University of Arizona, as an undergraduate. And I actually had an amazing time there. One of the great opportunities and advantages of being at a place like that is that it's primarily undergraduates. And so all of the resources are focused on undergraduates, which is what you are as a college student. And so I actually was able to enjoy the full brunt of the resources and do research in a lab for 2 years. I did another research internship for another year, and I got paid to do it because larger universities have programs that have funding to pay students to do research, which is a really big deal, especially in the economy and the way it is today. Trying to go through college and trying to find, because it's difficult to go through college if you're not getting some kind of income. And so that's a really helpful thing. So I'll make a plug for state universities and universities that typically have lower acceptance standards, but have exceptional education. As far as coming and going to different countries, I don't have a lot of experience with that at all at the undergraduate level, because I was born in the United States and I stayed in the United States. I did move states, but that's not as much of a move. I do know a lot of international students in the graduate program here, and they are very successful and they do a great job. And we have a really good support network for the international students at the university level. Visas are, as always, a difficult thing to get, but I think that if you can get it accepted into university, that will really help that going along. But I will end my advice there because I'm not an expert on this type of thing, so I really don't want to say anything that's not true. But I would definitely encourage, trying to reach out to universities and things like that. I also want to make a plug for, if you have, universities where you are, then that is also something that should not be overlooked. And I think that this is a kind of recurring theme of mine that, there's always the big famous top universities and everybody talks about those and everybody knows about those. And as a result, a lot of other universities kind of get overlooked. And so, I went to a university that I had never heard of before I started applying. My brother went to a university.